Hello, everybody, and welcome to this third webinar of this webinar series of the Open Brain Institute. So let me introduce myself. My name is Natalie Barros Zulaika, and I'm a scientist in this organization. And today I will walk you through this session that I entitled From Neuronal Morphologies to Insights using the OBI virtual labs. So as a start, let's first introduce what is this Open Brain Institute. So we are a non-profit organization, and we want to support researchers and organizations to build and explore digital models of the brain. We do that through environments that we call virtual labs. And in these environments, we have the computational modeling services. We also provide advanced tools, and we have repositories of digital brain models and data. And the idea is that people can implement realistic brain simulations, test their hypotheses, and explore the complexities of neuronal circuits. I want to emphasize that all our code is open source. All the data and the models are open source too. They are openly available, and they are totally free for you to try. But who are we? Who is these people? So we are a team of around 35 scientists, engineers, and designers based in Lausanne, in Switzerland. The institute was founded by Henry and Camilla Markran in January 2025. So we are a very young organization. Henry also founded at the EPFL, the École Polytechnique de Lausanne, the Blue Brain Project. That was a Swiss initiative. That project lasted for 20 years and was completed in December 2024. So the OBI is the continuation of the BPP, and the goal is to make available to all the community the models that we have developed for a long this time. So what are we gonna see in this webinar? So here what I want to show you is how can we use the platform to work with morphological reconstructed data. And I'm gonna explain that to you through 3 specific use cases. So we are gonna explore the data in the platform. I'm gonna show you how to upload your own data, test your data for errors, analyze the morphologies in 2 different ways, with common morphometrics and with a more complex topological analysis. And then I'm gonna show you some comparative analysis between species. So this is the specific content of the webinar. So first, because I don't know the audience, so maybe there are some people here that are new in neuroscience. So I'm gonna do a very basic introduction on neuronal morphologies, that what are they? So what are we referring to when we are talking about the neuronal morphology, and why are they important? Also, I'm gonna explain to you like what are these quality checks. And then we are going to see the first use case, which is check your morphologies for errors. Then we are going to see some of the analysis that we can perform in the OBI platform through a Python package called NeuroM. And then we are going to have the second use case, which is in case you want to complete your own data sets, you can use the data that we have in the platform. And then I'm going to show you a more complex analysis, which is called the topological morphology descriptor. And we are going to go to the third and last use case, which is comparing morphologies between species. And then, well, I will acknowledge you, and there is a round of questions, and so I will answer all your questions. And you can post your questions in the Q&A that you have in these Teams in the link. So you can post the questions there and I will answer them at the end of the webinar. So what do we mean by neuronal morphology? So neuronal morphology is basically the geometry of a neuron. So we know, I mean, a typical neuron is divided in 3 parts. So there is the soma. You have here in the figure, you have the nucleus in the soma. Then you have the dendrites, there is branching structures that function as the antenna of the neuron. So they receive incoming signals from other cells. And then there is the axon of the neuron that is specialized in the specialized out fiber that propagates electrochemical signals. So is where the signal goes out. So this is what we are talking about when we refer to neural morphologies. And why are they important? So they're important for several reasons. One of them is defining cell types. So in modern neuroscience, increasingly defines neuron types using combinations of morphology, transcriptomics, electrophysiology, connectivity. But morphology is one of the most robust descriptions of neuronal identity. And why do we need to define cell types? Well, it's important because like that we can map the brain. It improves the communication between the scientists, right? Because we can discuss about the different types that we are studying. And it helps also with the reproducibility of experiments and results. And here at the left, I just put an example of one publication in where they classify morphologically all the cell types that is found in the somatosensory cortex of the rat. So there is also the neural morphologies are important because they shape the electrical signal. So if we based our knowledge with the Rawls cable theory, we can say that electrical signals propagate through neural dendrites and axons as passive electrical cables. This theory explains how membrane resistance and axial resistance and membrane capacity shapes the spread and attenuation of voltage along neuronal processes, showing that dendritic morphology strongly influences signal propagation, synaptic integration, and neuronal computation. So in this example, there's an example of 3 morphologies, 3 pyramidal neurons in layer 2, 3, and in layer 5 from the rat visual cortex. And you can see that the 3 of them have very different electrophysiological properties. And this is due to their morphological shape. Neural morphologies are also important because they constrain the network connectivity and the network computation. So a neuron, as is obviously to think, can only connect where its dendrites receive inputs, axon reaches targets, right? Therefore, when connecting several neurons, as in a neuronal network, we could say that the connectivity of the network is constrained by the individual shapes of the neurons embedded in that network. But as neuronal shapes also affect signal propagation, that means that the propagation of the signal within the network would be affected by the shapes of the neurons forming the network. And as a consequence, it will affect also the computation of the signal within the network. So here I want to show you an example of this study in which the authors built 2 models, 2 networks of mouse. One network was mouse with pyramidal neurons, and another one it was human with pyramidal neurons. And they saw that the complexity of the human network was higher comparing to the complexity of the mouse network, and that complexity came from the shape of the morphology. And they saw that the human neurons, they had a more complex branching around the soma comparing to the mouse networks. And they also predicted that a network built from neurons with more complex dendritic patterns would be a network with an increased memory capacity. And of course, the fact that our morphologies are not, don't have a correct shape, so to say, also provokes leads to illness. So there are many neurological and psychiatric disorders involved with structural abnormalities of neuronal morphology. So, for instance, we have dendritic atrophy in Alzheimer's disease. There are altered spines, then the density in schizophrenia, axonal degeneration in Parkinson, or developmental defects in autism. And here we have an example of a study that is comparing healthy, normal patients with schizophrenic patients. And you can see that the neuronal morphology of these patients, the schizophrenic ones, are different, right? The area of the soma is decreased, or most of the parameters are decreased, but the axonal length was increased. And not only in the shape of the morphology, but in the shape of the spines. So you can see here, this is the total number of spines and the different type of spines. In the case of the schizophrenia patient, the number of spines was reduced. So now that I think it's settled, that morphological reconstructions are important for us, for the people that study the brain, I'm going to tell you a little bit broadly, very and briefly, how can we get this data? So the process goes more or less like this. So in the lab, after some steps for preparing the tissue, sometimes experimenters, they label the cells that they want to study, they target them, they can stain them, they can use viruses. There are different techniques. And after that, what they do is they take pictures, they slice the brain, they take pictures of the brain, and then they study this set of pictures. And there is also the possibility of having a block of tissue and then taking very, very precise and specific pictures while slicing the brain. And this is called the electron microscopy images. And with this set of images, people, they use different softwares, and then they can reconstruct the whole or most of the morphology in 3D. And then this reconstruction basically goes into a file that they can be read by a computer. So at the Open Brain Institute, we support three types of files, three formats: ASCII, SWC, and H5. And we do that because we consider that these three types of files are the ones that are more used in the community. And some concepts that I want to introduce you to, in case you are not familiarized with Neuron. Neuron is a simulation environment that we use in the code that we use in the Open Brain Institute in our platform. And there are 2 points when studying morphologies that two concepts that are important is the concept of section and the concept of segment. So a section is the continuous biological cable structure. So you have here in the picture on the right, this is a segment. It could be like a piece, whole piece of dendrite. It could be a soma, one dendritic branch, one axonal branch. However, the segment is a subdivision of this section. So a section has geometry, has a length and a diameter. It represents an unbranched cable. However, the segment is only a subdivision of the section. And we need it because we use Rolle's cable theory equation. So the sections need to be discretized. This is just you to keep you in mind once you go to the code and in case you want to modify the code. This is important to understand. But then, okay, so after you have the reconstruction, well, reconstructing neural morphologies is a very complex task. So some errors could happen during the reconstruction. Some of these errors are not really important if you just want to study the shape of the morphology. However, if you want to use this morphology for simulation, these errors are very important. So here I put a list of the 6 most important errors that we consider that are most important. Like basically they are disconnected branches, right? We have the zeta jumps, we have a dangling branch, or in the case that they are zero or soma or neurites, or also they need to be 3D. So if we have flat neurites or the shape, right? We know that branches go from soma to the end, like they go from thicker to narrower. If the shapes of the sections or the thickness of the sections go inversely, or there's something weird. I mean, the thing has always to do with the flowing of the signal through the cable. So if the cable is wrong or there is something broken, then it won't go properly. So your simulation will be wrong, basically. So we need to fix these morphological reconstructions in case there are some errors like those. And for that, we have a code, and we provide a code in the OBI platform, so you can test your morphologies for this type of errors. Our code also check for missing neurites. This is not by itself an error, right? Because, for instance, there are 3 examples here. Most of the morphological reconstructions, they don't have axons, because they are very, very hard to reconstruct, but still you can do a lot of research with just the basal and apical dendrites. So this is not a mistake, but it's also information good. I mean, it's good to have, to know that your morphology, for instance, doesn't have a reconstructed axon. Or even in biology, we can find morphologies without basal dendrite, like the Purgine cell, or morphologies without apical dendrite, like the basket cell. So let's go to our first use case. So imagine that you're an anatomist or you are a modeler, and you're collaborating with an anatomist or a modeler. Okay. So the idea is you have received a set of data, or you already have reconstructed them, and you want to put them in the platform, and you want to test either some errors in these morphologies because you want to use them for modeling. So we are going to see how to upload your data and how to run the check morphology notebook. So if I go to the platform, yeah. So this is our homepage. You can see here, for instance, my project name, my virtual lab name, the credits, but let's focus on the use case, okay? So if I go to data, you can see that we have 2 different types of data modality. We have the public, is data that is available for everybody. Everyone that wants to access the platform has access to this data, or we have the project data. In this case, you see I have 7 morphologies for rat because I uploaded rat morphologies. And this data is only available for the people authorized in the project. So only for the people working in this project. So it's private data. So if I go here, you see I selected all this, but I'm going to show you that after. And I want to upload one of my morphologies. So I go here on the right, you click on the green button, upload data. The first thing to do is to select what type of modality do you want to upload. So okay, let's say morphology. Then I go to my files. I already have some morphologies here. So I'll uploaded something. I'm gonna upload, for instance, this one. Okay. So immediately our system, what it does, as we support the 3 types of data format, it converts your file into the other 2 data formats. So you already have the 3 data formats there saved in your project. So I click on the next arrow, and then I have to introduce some metadata, right? So let's see, I was rat morphology 09, if I remember correctly, is pyramidal neuron from layer 3 cortex, whatever. So the brain region, I said it was primary somatosensory barred field. Experimental day, this is not mandatory, let's say today. Contact email is not mandatory neither. Publish or location. Now, sometimes it's important to have the locations of the morphology. I mean, if you're lucky and you have this data, you can register it in the platform. The protocol. Let's say a generic protocol. Let's say is raw data. So this is the person who registered, is me. I am a data modeler, and this is my name. I mean, it's there because I already registered many data, and you can add more contributors in case you have collaborators with you. And the subject is just let's see, general. Here, sorry, overage rat. And the license I'm gonna keep non-commercial. And I say it was layer 3 beta million. Okay, I confirm. Okay. Sometimes it can take a while, but after a few minutes, you will see this circle becoming green. So you will have it there. And for the sake of the webinar, I already have, you see, view details. Perfect. Okay, so here you see this is the detail page of the morphology that we just uploaded. You can zoom in, zoom out on the here in this detail view. You can rotate, you can double click and have full view, and you have all the metadata that we just included in the registration. And on top of that, you have already some morphometrics computed for the morphology that are also registered there. So you can download, you can delete it, or this copy ID is to use this data in a notebook. So, okay, as I showed you before, I have, and you have them, they're here, all of them very beautiful. But as we said before, we want to check if these morphologies are useful or they can be used for being simulated, for being used in a model. So I have my notebooks here in my project. So similar to the data, you can also upload notebooks in your project, or there is also a big list of notebooks that we provide. These are public and everybody can run them. And the ones in my project are going to be public too after the webinar. So you can use them and run them to check your morphologies for errors, for instance. So we go here, you have run morphology error check, there is some description on the notebook, and we just click run, and then it will take us to the Jupyter notebook environment. Yeah. Okay, so let's run the cell. So in case you are not familiarized with the Jupyter notebook, it's very easy to run. You see the cross here, the cursor. You select the cell. You can just click play, or in your keyboard press Command Enter, and then the cell will run. So the very first thing you need to do is to authenticate yourself. So yes, this is me. I'm authorized to run this notebook. Then, like, okay, select your project, the project where you want to run the analysis. The client, these are some Python packages that we need for the analysis. For the notebook, I said we had 8 morphologies. Let's check 7. And you just keep running. So right now it's basically downloading the morphologies that I have in the project. Here, you see? You can see the folder here. Yeah. So now the morphologies are accessible through the notebook. So yes, keep running. And here, basically, it's a widget, and you can select the morphology list. So for instance, let's check the first one. Okay, there is one error. What is the problem? Okay, there is a dangling branch. Okay, there's a problem. There is one branch that is disconnected. So let's check the one that we just uploaded. Oh, this one is perfect. Oh, no. There is something. What's happened? As axon, true. Non-zero segment length. There is one segment that is 0. So usually what we do is to send these morphologies back to the lab to be corrected, but then you can actually point to the experimenter in the lab what are the issues with the morphologies. And in the future, we are preparing a repairing pipeline for the morphologies that have these issues, but it's still to come. So if I go back to my presentation, okay, so we have learned how to upload and how to check morphologies for errors. Now, well, let's analyze them, right? So what are the options that we are offering at the Open Brain Institute for analyzing morphologies? So we have this Python package called NeuroM that was developed in the epoch of the Blue Brain project, and will allow you to perform morphometrics. So what is what is morphometrics? So morphometrics is a quantitative analysis of the geometry of the morphology. So what can you do with NeuroM? You can read and parse the neuronal morphology. It can read these three formats, ASCII, SWC, and H5. And you will use Python code. Then you can compute the morphometrics like section length, branch angles, radial distances, a lot of them, with a single functional cell. And you can work natively with the tree structure of the neurons. So you can actually say soma, axon, basal dendrite, or apical dendrite without needing to use complicated code. What it facilitates? So as I said, you can extract a lot of morphological features just with one line. You can analyze a whole population. You don't need to go one by one. You can visualize the morphologies within the package, and there are also validation tools. This is a complete list of all the morphometrics that you have available in the package. I'm not gonna go through all of them, but you see there are a lot. You have the link in the notebook that I'm gonna show you right now to the NeuroM GitHub repo, so you can check there what each of the analyses means. But let's work now on our second use case. So here you can see my virtual lab again. If we go to notebooks in my project, you can check that there is a notebook called complete dataset. We go there, we click run. And again, we are in the environment of the Jupyter notebook. So the very first cell, as we did previously, to load some packages and the access token. As I already I'm authenticated. I don't need to do it again. I select the project where I'm gonna work into, and I ask for client name and number. I upload some packages, and then the very first thing that what we are gonna do is to upload in the Jupyter notebook some of the morphologies that we have uploaded in our previous use case. And let's see that I'm gonna check 5, and then I upload them in this Jupyter notebook environment. And then you can see here on the left how the folder morphologies have been created, and here I have 5 of them. We can also perform some analysis on them. Here I just selected 2 random morphologies, and for instance we can plot the dendrogram. We can see that there are very complete reconstructions, and also you can see that even though reconstructing the axon, which is these blue lines, is very hard, these morphologies, they have a very complete axonal reconstruction, so they are very nice. We can also compute the section lengths. We have a distribution, for instance, and this is what we are going to compare with the morphologies in the platform. So now we're going to look for the morphologies in the platform. And the first thing that we should do is to select the species. So my morphologies are rat. So I select rat. The brain region, they are from the primary somatosensory cortex. So I'm going to look for this primary area, highlighting representation of layer 2-3, because the morphologies are layer 2-3 pyramidal neurons. and then I look for the cell type. This one, layer 3, GPCA, and I upload them. Also, I'm uploading 5. Okay, so now if I check in the folder, you can see that there are the morphologies in my project plus 5 other morphologies from the platform. And of course, I mean, if I want to do some studies with those to complete my data set, we should check that they are comparable. So let's, for instance, compare the section length of the basal and apical, so of the all neurates, basal, apical, and the axon. And you can see in color these are the morphologies from like the dataset that I uploaded, and in black are the morphologies from the platform. So you can see that the distributions are pretty comparable. However, of course, I mean, as I said before, morphologies that I uploaded have an amazing reconstruction, so the number of sections are bigger. However, the ones in the platform, they don't have as many number of sections, but the distributions are comparable, so we would be able to complete our data set with these morphologies that I selected from the platform. Now let's back to the presentation. So let's say that you want to perform more complex analysis than just simple morphometrics. We provide this notebook called that performs this analysis, the topological morphology descriptor. I want to use this moment to acknowledge Lida Canari here in the picture. You can see her, who is a researcher fellow at the University of Oxford, and she has been working and collaborating with us for many years. She was our expert on morphologies, so none of this work that I'm showing here would be possible without her. So thank you very much, Lida. So what can you do with TMD? So the core idea is to use topology instead of metrics. So the TMD algorithm encodes the branching pattern of the morphology by discarding local fluctuations with little information content, such as the position of nodes between branch points, reducing the computational complexity of a tree. This makes it robust to noise and reconstruction artifacts. You can use it to compare neuron shapes mathematically using TMD distant metrics, which is useful for ranking how similar two morphologies are. Or you can use it to rank automatically reconstructed neurons against a reference morphology, for instance, or to classify morphologies. So the idea is that TMD provides with a fingerprint of the shape of the morphology. And we also have a Python package for TMD. So in a schematic way, to show you how this works. So from a dendritic axonal tree that you have here, it extracts a barcode. The barcode is based on the distance between the soma and the end point of the bifurcation of the tree. For each neuron, one barcode is generated, and each barcode is unique. However, neurons that belong to the same type, shape-wise morphologies, will share similar characteristics. These characteristics can be better observed in the persistent diagram and the persistent image that belong to the barcode. So here you have an example of a human morphology. From this dendritic branch, you can extract the barcode on the right. And then below, you have the persistent diagram. In the persistent diagram, each pixel's x-axis represents the distance to the soma. And its distance from the diagonal it represents the length to the branch. And in the persistence image, each pixel's color intensity is determined by the density of the branches at that location. That results in an image summary of the topological complexity of the branching pattern. And this image-based topological summary has been used in several applications, such as neuronal morphology classifications, as I told you before, and also glia classifications. So we are going to use it to find differences between species. And this, just to mention that this analysis has been also used as a mathematical method to synthesize morphologies, which means that these neurons, these morphologies, have been grown in the volume, and they have grown with these certain characteristics that comes from this analysis. And you can find this type of data in our platform. So let's go, well, before going to the last use case, here is a table just comparing NeuroM to the TMD analysis. So what do we get from NeuroM? Basically, the measurements like morphometrics, and the TMD, we get the topological fingerprint. NeuroM is best for descriptive statistics and for plotting, and TMD is for classification, comparison, and clustering. And NeuroM is based on classical morphometrics, and the TMD is based on persistent homology. So let's go to our last and third use case. We are going to run a notebook with morphometrics, and then run a notebook with a TMD. And we are going to show you some results. I go to the platform, I go into notebooks, okay, compute morphometrics. Yeah. So I'm gonna run it fast because we are, okay. Let's find the project. That's it. Yeah, you have here the links to Neuromorpho in case, to NeuroM, sorry, in case you want to check the code. Now what we're gonna do is to find 20 morphologies from mouse, 20 morphologies from rat, and 20 morphologies from human in the platform. Yeah. You will see the morphologies appearing here on the left in the respective folders. We can load them here. And then, for instance, we can plot one of them for each species. Yeah, you have them here. You can also plot the dendrogram. Yeah. And this is the list of all the analyses that you can run. I leave it there for you to check. You can also get data frames of the analysis, and you can also compare them here. This is human versus mouse. And for instance, let's compute the section length for these morphologies of the basal dendrite. Okay. You have in red the mouse, the rat in purple, and the human in teal. And you can compare distributions. You can see here, of course, rat versus rat. There's no difference, but there are already differences between rat, mouse, and rat human. So here in the next cell, if you run it, what you will get at the bottom is a widget with a full list of the analyses. If you click in one of them, let's say that we want to do soma, for instance, soma volume. And here you can see, well, this rat neuron is huge comparing to the human neuron. That sometimes happens. And this is the mean of all the 20 neurons from each species. For instance, we can select the section radial distance, and we get a distribution of all the radial distances of the sections for each morphology. Yeah, we have, these rat morphologies are pretty big. You can see. Okay, so this is the morphometrics. But what if we want to run the TMD, the topological analysis? So we will go compute topological analysis. We go here, run. And the notebook gets open. And we run it again. Selecting project. We will do the same. We will select 20 random morphologies from mouse, 20 random morphologies from rat, 20 random morphologies from human. We see that they have downloaded here at the left. They are here, there in the notebook environment to be used. So similarly to before, and using the TMD package, we can plot some of the morphologies. Yeah, a human, rat, a mouse. This is the tree for the human neuron. You can see that you can compute the persistence barcode, the persistent diagram, and the persistent image. Let's see the topological barcode. This is for one neuron selected randomly from each of the sets of data. The topological diagram. You see the rat morphology has a very interesting shape comparing to the other ones. And now here we have the top of the persistent image of the rat morphologies. This is the overage. And the mouse and the human. So you can see that they are very different, right? Already. And we can also, this is for just one morphology, and we are going to compare now the difference between them. You can see here is the difference rat versus mouse. So here there is more rat, here there is more mouse, so to say. The blue, the negative values. So you can see that the differences between these two data sets is that there are a lot of density of branches here, around 400 micrometers from the soma for the rat, and around 150 micrometers for the mouse. And you can normalize that also for the human and for the mouse, for instance. In this case, you can see that the human has more, they're not symmetric, right? You can see that. Well, this is just a random, of course. And there is another study, another analysis, that you can perform, which is the entropy, which that shows you also is another measurement of complexity. So here you have the mouse and the rat. You can see that the complexity of the rat is not near the soma, the 0 is the soma, is like in the middle of the apical tree. So with that said, what have we learned? So technically, we have learned how to explore data in the platform, how to upload data, or mostly morphological data in the platform, how to run the notebooks and the analysis on the notebooks. And I want to emphasize that these analysis and everything that we have in the platform is openly available and that is free for testing. And specifically, we have checked for reconstruction errors. We have performed morphometric analysis and topological analysis, and we have compared data sets between species. So in the near future, we will make available the capability of uploading several morphologies simultaneously, not one by one, like a set of a group of morphologies. And we will provide also a repair pipeline. So you can repair your morphologies and you don't need to go back to the lab. I also encourage you to check our previous webinars. They are available in our webpage or in our YouTube channel. There is another new webinar coming soon, and it's entitled Pharmacological Modulation in Silico with Genetic Ion Channel-Based Single-Cell Modeling. And I encourage you to subscribe to our newsletter, where you will get all the news from our webinars. And I really want to thank you for your attention, and I hope you enjoy the webinar. Yeah, now there are some questions. Okay, so from Duanga Tai, I'm sorry, I hope I'm telling your names properly. Do you have any platform or data set that can analyze the neuronal cell culture? Can we upload the data, pictures, and analyze the morphology, branching, density, length, etc.? So what you can upload is, I'm not sure what you refer to, data pictures. So you cannot upload the stack of like the picture itself. You need to upload the type of file, like as I said, or RC, SGBC, or H5, so the morphological reconstruction. But any morphological reconstruction, it doesn't matter where it comes from, it can be analyzed in the platform. Also, will we be able to analyze several neurons at the same time from one frame, as the data derived from the neuronal cell culture? I mean, you can analyze as many neurons as you want. We have the capability through the notebook, you can analyze all of them. Can we utilize this to build the human connector that represents all the connections in human brain?
In principle.
In principle, yes.
You can predict a connectome from a photo.
Yeah, exactly. So we are releasing a paper actually about that. Yeah, you can build a connectome with the human morphologies. We will also make available a synaptome. I'm preparing also, we have a human cortical model, and it will release hopefully at the end of the year. So yeah, but anyways, I mean, with these morphologies, you should be able to, in principle, to get a human connectome.
But for all connections, you would need a lot of morphologies.
Yeah, that's also true. So the main issue is always the lack of data. Yeah, you're welcome. There was another question.
Just following up on the last question from the previous person about the having the entire human connectome, theoretically doing analysis on the entire connectome. I know this is probably not directly relevant towards developing new morphologies and analyzing morphologies, but in theory, if you're dealing with very in the future, is there a roadmap towards integrating, say, for example, very rich or channel dense EEG sets for example, maybe 120 channels plus, and you're kind of performing source analyzation or connectivity metrics and stuff like that. So is there a way to on the very distant future roadmap of kind of integrating live data sets with the kind of models that the open brain, what your institute has been developing, so that you can actually sort of play around with these things in real time using real-time data sets? That's my question, but I'm not sure if it's very related to what
I mean, I have here at my left like the scientific chief of the OBI, and he is an expert in connectomics, and he is like pushing me like, I want to answer this. So
yeah, sorry, let me just take the question. Hi, I'm Michael. And short answer, yes, like on our roadmap is also representation of EEG data and eventually even simulation of EEG data and providing an analysis platform for EEG data. But it's not the near future. We have a lot of other stuff to deliver, but it is definitely on our roadmap. So if you stay subscribed to our newsletter, you will be updated once it's available.
Okay, thank you for that. And just as an adjacent question to that, do you think if people were in the space of creating very electrode or channel-rich data sets, would that be actually beneficial towards this integration, or not really, or it doesn't quite matter because
I think it's going to be amazing for as a data set for the validation of simulations. That's where I'm really keen on having this. Just imagine you have you're really branching out. You're building a multi-region, maybe whole brain or just large parts of the brain data set. You need something to validate against the emerging activity, and this kind of data would really just be invaluable because to get the EEG signal right, there are so many individual parts you need to get right. It's very invaluable.
Okay, thank you so much for answering the questions.
Great. So I'm handing over back to Natalie.
Thank you so much for attending. I mean, if in any case you have questions or further questions, you can always write us an email. Thank you. Bye.
Bye.